Right. So as you know already, dear students, concerned with the first chapter, I covered 99% of the matter. Concerned with first chapter, I covered around 99% of the matter. Left only one matter is sexuality in organisms. Sexuality in organisms. Previous class, I come across introduction to second chapter, sexual reproduction in flowering plant. Under that, I covered definition of flower. Followed by that, I come across a typical flower is composed of four parts: calyx, corolla, androecium, gynoecium. Concerned with androecium and gynoecium, I covered introduction. Members of the androecium are called as stamens. It is composed of two parts, anther and filament. Gynoecium is the fourth and it is the innermost floral oral. Members of gynoecium are called as carpel, and each carpel is composed of three parts: ovary, style, stigma. Then I discussed post-fertilization changes in flowering plants. After fertilization, sepals, petals, stamens, withers, and they will fall down. Sepals, petals, stamens, withers, and they will fall down. Zygote will develop into embryo. After fertilization, ovary will develop into fruit, ovules will develop into seed. After dispersal, seed will germinate and produces plantlet. Fruit is covered by a wall known as pericarp. Followed by that, left matter is sexuality in organisms. Under that, you are going to study bisexual, unisexual. Under unisexual, two types: staminate, pistillate, monoecious, dioecious, and in animal also you will get unisexual and bisexual condition. Okay, under sexuality in organisms, you are going to study the technical terms like bisexual, unisexual, monoecious, dioecious. Followed by that, I will introduce animal matter. Followed by that, you are going to study continuation part of second chapter, sexual reproduction in flowering plants. When a flower possesses, or when the flower is having both androecium and gynoecium, then it is called as bisexual flower. When the flower possesses both androecium and gynoecium, then the flower is described as bisexual. Bi means two. When the flower is having both androecium and gynoecium, then it is called as bisexual. Unisexual, flower is having only one essential oral. Previous class we have come across out of four floral orals: calyx, corolla, androecium and gynoecium. Lower two floral orals, that is calyx and corolla, are called as non-essential floral orals because they are not directly involved in sexual reproduction, whereas androecium and gynoecium are called as essential orals because they are directly involved in sexual reproduction. Unisexual means the flower is having only one essential oral. It may be an androecium or gynoecium. They are further classified into two groups, staminate and pistillate. Staminate means male flower. Staminate means male. Pistillate means female. Staminate means it is having only androecium. Pistillate flower means it is having only gynoecium. When flower is having both androecium and gynoecium, then it is called as bisexual. When the flower is having only one essential oral, when the flower is having only one essential oral, then it is called as unisexual. Under that, two types, staminate and pistillate. Staminate means male flower, it possesses only androecium. Pistillate means female flower, it possesses only gynoecium. Monoecious. When staminate and pistillate flowers, when staminate and pistillate flower are present on the same plant, then it is called as monoecious. When the plant is having staminate and pistillate flower, then it is called as monoecious. Example is coconut and cucurbits. When staminate and pistillate flowers are present on the same plant, then it is called as monoecious. Example is coconut and cucurbits. So please remember one line. Coconut is commonly called as Kalparuksha in Karnataka and Kerala. Coconut is commonly called as Kalparuksha in Karnataka and Kerala. Dioecious means sexes are separate. Simple meaning of the dioecious is sexes are separate. When staminate and pistillate flowers, when staminate and pistillate flowers are present in different plants of the same species, then it is called as dioecious. When staminate and pistillate flowers are present in different plants of the same species, then it is called as dioecious. Example for a dioecious is papaya and date palm. When staminate and pistillate flowers are present in different plants of the same species, then it is called as dioecious. Example is papaya and date palm. Papaya and date palm. Next matter is with respect to animals. Animal may be unisexual or bisexual. Like plants, animal may be unisexual or bisexual. Unisexual example is cockroach. Animals may be unisexual or bisexual. Unisexual example is cockroach. Classification and explanation I come across in PUC first year chapter structural organization in animals, phylum Arthropoda, class Insecta, order Dictyoptera, family Blackidae, genus Periplaneta, species Americana. It is a native of America, but now they are found all over the world, including my great India. Bisexual, alternate technical term which is used is hermaphrodite. Bisexual is also called as hermaphrodite. When both male and female reproductive structures, when both male and female reproductive organs are present in the same individual, then it is called as bisexual. When male and female reproductive structures are present in the same individual, then it is called as bisexual or hermaphrodite. Many examples that are quoted in your NCERT book, including leeches, sponges, earthworm, tapeworms. When Both male and female reproductive structures are present in the same individual, then it is called as hermaphrodite. Example is Indian cattle leech, sponges, earthworms, tapeworms. So that ends the complete matter related to first chapter. Already under second chapter I covered definition of flower, thalamus, parts of flower, introduction to androecium, introduction to gynoecium I covered. Now look over an important 5 marks matter, transverse section of a young anther. 5 marks matter, transverse section of a young anther or microsporangium. Now I have to take care how to draw the figure. First you make an rough outline, followed by that you dark it. Epidermis, just below that one, endothecium, endothecium, middle layer, middle layer, tapetum, this sporogenous tissue. This is a transverse section of young anther. Transverse section of young anther. This middle part is connective. So in your NCERT, they are given both the figures. So this figure, along with that, an expanded figure also they are given to draw both of the figures in the annual examination. Okay. Okay. Now look over important 5 marks matter. So dear students, to avoid the loss of time and your internet, I drawn a rough figure. Okay. So this and transverse section of an young anther, or you can call it as a structure of microsporangium. The question will appear for 5 marks. At that time, figure will carry 2 marks or sometimes 2.5 marks, and explanation will carry 3 marks or 2.5 marks. Okay. Now look over explanation. I will explain the matter from periphery towards the center. I will explain the matter from periphery towards the center. This is an ground plan of the TS of young anther, out of which they expanded only 25% of the figure. Okay. Now look over the lining. Epidermis. The name epidermis is derived from Greek. The name epidermis is derived from Greek. Epi means up on, derma means layer. The name epidermis is derived from Greek. Epi means up on, derma means layer. So epidermis is the outermost layer. It is made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Epidermis is the outermost layer. It is made up of barrel-shaped cells. Cells are tightly arranged or compactly arranged without any intercellular space. Main function of the epidermis is to provide protection. Instead of this big explanation, you can reduce the line. Epidermis is the covering tissue. It gives protection. Just below the epidermis, endothecium is present. Just below the epidermis, endothecium is present. It is made up of single layer of radially elongated cells. It is made up of single layer of radially elongated cells. Radius, you know, in mathematics, it is a straight line from center to the circumference of a circle. According to Mahesh sir, it is a straight line from center to the circumference of a circle. So just below the epidermis, endothecium is present. It is made up of single layer of radially elongated cells. Endothecium cell is having fibrous thickening. It is having fibrous thickening. That is nothing but deposition of cellulose and little pectin and lignin. Just below the epidermis, endothecium is present. It is made up of single layer of radially elongated cells. Endothecium cell is having fibrous thickening. That is nothing but deposition of cellulose and little pectin and lignin. So this layer will play a significant role in the splitting of the anther to release the pollen grains. Endothecium will play a significant role in the splitting of anther or in the dehiscence of the anther, to release the pollen grains. Inner to endothecium, middle layer is present. It is made up of 3 to 4 layers of, thin-walled cells. Just below the endothecium, middle layer is present. It is made up of 3 to 4 layers of thin-walled cells. Inner to middle layer, tapetum is present. Epidermis is the outermost layer, tapetum is the innermost layer. It is composed of dense cytoplasm with one or more nuclei. Tapetum is the innermost layer of the wall of young anther. It is having dense cytoplasm with one or more nuclei. Tapetum is a nutritive tissue. It will supply the nutrients to the developing pollen grains. Epidermis is the outermost layer. Tapetum is the innermost layer. It is having dense cytoplasm with one or more nuclei. Tapetum is a nutritive tissue. Tapetum is a nutritive tissue, and it will supply the nutrients to the developing sporogenous tissue. It will supply the nutrients to the developing sporogenous tissue, or you can call it as a future pollen grains. Okay, so that ends the transverse section of a young anther. But you have to remember one more tissue. When the anther is young, okay, when the anther is young, you will get a group of 4. 1, 2, 3, 4. When the anther is young, you will get a group of 4 compactly arranged homogeneous cells known as sporogenous tissue. When the anther is young, you will get a group of 4. 1, 2, 3, 4. Group of 4 compactly arranged homogeneous cells called as sporogenous tissue. Group of 4 compactly arranged. Compactly means tightly arranged. Homogeneous means same type. Homogeneous cells called as sporogenous tissue. As the anther develops, sporogenous tissue will undergo meiotic division and produces pollen tetrads, or you can call it as a microspore tetrads. These microspore tetrads are get separated and develop a characteristic shape which differs when comparing with other species. As the anther develops, sporogenous tissue will undergo meiotic division and produces microspore tetrads. These microspore tetrads are get separated and develop a characteristic shape which differ with respect to other species. Next matter is structure of pollen grain. Before that, remember one important line: the process of formation of microspores, the process of formation of microspores from pollen mother cell is called as microsporogenesis. The process of formation of microspores from pollen mother cell by meiosis is called as microsporogenesis. Okay, so I hope that my explanation is clear. Epidermis will give protection. Endothecium will play a significant role in the splitting of the anther to release the pollen grains. It is having fibrous thickening which is composed of cellulose and a little pectin and lignin. Tapetum is the innermost layer. It will supply nutrients to the developing pollen grains. Okay. Next is structure of pollen grains. So to avoid the loss of time, I'm drawing the rough figures. Once please go through NCRT figures. Okay. Once go through NCRT figures. Structure of pollen grain or microsporangium. I will repeat the previous point. When the anther is young, you will get a group of 4 compactly arranged homogeneous cells called as sporogenous tissue. As the anther develops, sporogenous tissues will undergo meiotic division and produces microspore tetrads. These microspore tetrads are get separated and develop a characteristic shape which differs with respect to other species. The process of formation of microspores from microspore mother cell by meiosis is called as microsporogenesis. Now look at one more important matter, structure of pollen grain or microsporangium. It will appear generally for 3 marks. Okay. So pollen grains are spherical in shape. Spherical means ball-like. Pollen grains are spherical in shape, measuring around 25 to 50, measuring around 25 to 50 micrometers in diameter. Pollen grains are spherical in shape, measuring around 25 to 50 in diameter. So take care of the lining. Pollen grains are spherical in shape, measuring around 25 to 50 in diameter. It is covered by an wall which is made up of 2 layers. It is covered by a wall made up of 2 layers. Outer is called as exine and the inner is called as intine. It is covered by an envelope or wall which is made up of 2 layers. Outer is called as exine and the inner is called as intine. Exine is tough. Exine is tough. Cutinized. Exine is tough. Cutinized. Cutin is present. So this word cutin we have come across in PUC first year, anatomy of flowering plants. Outer wall of the epidermis, back to first year, chapter morphology of flowering plants. Outer wall of the epidermis will show a thin waxy coat known as cuticle. Cuticle not only gives protection but also checks the rate of transpiration. So here, exine is tough, cutinized, cutin is present, often means sometime it is provided with spinous outgrowth. Often it is provided with spinous outgrowth. Now question arises, what is the use of this spinous outgrowth? So spinous outgrowth helps in the dispersal of pollen grains. Look over here. Pollen grains are spherical in shape, measuring around 25 to 50 in diameter. It is covered by a wall which is made up of 2 layers. Outer is called as exine and the inner is called as intine. Exine is tough, cutinized, often provided with, sometimes it is provided with spinous outgrowth. The spinous outgrowth helps in the dispersal of pollen grains. It helps in the dispersal of pollen grains. I will discuss this matter in detail later under entomophily. Pollination by the agency of insect is called as entomophily. It is tough, cutinized, often provided with a spinous outgrowth. Exine is having sporopollenin. So you have to remember this point for neat purpose. Exine is having sporopollenin, one of the most resistant organic material. It can withstand high temperature, strong acids and alkalis. Exine is having sporopollenin, one of the most resistant organic material. It can withstand high temperature, strong acids, and alkalis. Due to this presence of sporopollenin, pollen grains can be best preserved as an fossils. Exine is having sporopollenin, one of the most resistant organic material. It can withstand high temperature, strong acids, and alkalis. Due to presence of sporopollenin, pollen grains can be preserved as an fossils. Okay, so this word fossil we have come across in 10th standard. So as a part of my duty, I will provide you one simple definition. Fossils are the preserved remains of plants and animals. Fossils are the preserved remains of plants and animals that once existed. A beautiful example is dinosaurs. They are dominate during Jurassic era. Okay. Fossils are the preserved remains of plants and animals that once existed. Due to sporopollenin, pollen grains can be well preserved as an fossils. Interesting point. Enzymes will not degrade sporopollenin. Enzymes will not degrade sporopollenin. ನಾನು ಏನು ಹೇಳ್ತಾ ಇದ್ದೀನಿ ಅಂತಂದ್ರೆ ಈ ಎನ್ಸೈಮ್ಸ್ಗಳು ಸ್ಪೋರೋಪೊಲಿನಿನ್ ಅನ್ನು ಡಿಸ್ಟ್ರಕ್ಷನ್ ಮಾಡಕ್ ಆಗಲ್ಲ. ಹಾಗಾಗಿ ಡೈಜೆಸ್ಟಿವ್ ಟ್ರ್ಯಾಕಿನ ಒಳಗಡೆ ಎಂಟ್ರಿ ಹೊಡೆದ್ರೆ ಸೀದಾ ಅದು ಹೊರಗಡೆನೆ ಬಂದ್ಬಿಡುತ್ತೆ ಎಕ್ಸ್ಕ್ರೀಟರಿ ವೇಸ್ಟ್ ಮಟೀರಿಯಲ್ ಜೊತೆಗೆ. ಓಕೆ, ಸೊ ಎನ್ಸೈಮ್ಸ್ ವಿಲ್ ನಾಟ್ ಡಿಗ್ರೇಡ್ sporopollenin. So next is intine. Just below exine, intine is present. Just below the exine, intine is present. It is thin, delicate, and made up of only cellulose. Just below the exine, intine is present. It is thin, delicate, and made up of only cellulose. Now we have to take care some of the important CET points related to pollen grains. In around 60% of the angiosperms, of the angiosperm, pollen grains are released to the environment at a two-celled stage. Larger is called as vegetative cell and the smaller is called as generative cell. In around 60% of the angiosperms, pollen grains are released to the environment at a two-celled stage. Larger is called as vegetative cell, or you can call it as a tube cell. Smaller is called as generative cell. Vegetative cell is having abundant food. It is having more reserve food material with an irregular shaped nucleus. In around 60% of the angiosperm, pollen grains are released to the environment at a two-celled stage. Larger is called as vegetative cell and the smaller is called as generative cell. Vegetative cell is having abundant food material with an irregular shaped nucleus. It is having an irregular shaped nucleus. Generative cells are spindle shaped. Please remember, for CET purpose, generative cell is spindle shaped. Spindle means it is broad at the middle and gradually tapers towards the tip. Spindle shaped means it is broad at the middle and gradually tapers towards the tip. So generative cells are spindle shaped with a dense cytoplasm and nucleus. So I will repeat this point and take care. In around 60% of the angiosperm, pollen grains are released to the environment at a two-celled stage. Larger is called as vegetative cell, smaller is called as generative cell. Vegetative cell is having abundant food with an irregular shaped nucleus, whereas generative cells are spindle shaped with a dense cytoplasm and nucleus. It is generally floating in the cytoplasm. It is floating in the cytoplasm of vegetative cell. Generative cells are spindle shaped and they are floating in the cytoplasm of vegetative cell. In rest, around 40% of the angiosperm, pollen grains are released at a three-celled stage, where generative cell will undergo division and produces two male gametes. In rest cases, generative cell will undergo division and produces two male gamete before they are released into the environment. Before they are released into the environment. Pollen grains are rich in nutrients. Pollen grains are rich in nutrients. Therefore, nowadays it is fashion to consume pollen tablet as a food supplements. Pollen grains are rich in nutrients. So nowadays, our modern times, these pollen tablets are consumed as a food supplements. It is believed that it will increases the performance of athletics and even race horses. Pollen grains are rich in nutrients. So modern peoples or in modern days, pollen tablets are consumed as a food supplements. It is claimed that or it is believed that it will increases the performance of athletics and even race horses. Pollen grains can be well preserved or it is preserved months together in liquid nitrogen at minus 196 degree centigrade. Pollen grains are preserved in liquid nitrogen. In liquid nitrogen at minus 196 degrees centigrade. So this preserved pollen are used as an pollen bank. They are used as an pollen bank in crop breeding programs. Pollen grains are preserved months together in liquid nitrogen at minus 196 degrees centigrade. Preserved pollens are used as a pollen bank in the crop breeding programs. They are used in crop breeding programs. Last point. Viability of the pollen grain. So viability of pollen grain is controlled by temperature and humidity. It is controlled by temperature and humidity. In case of wheat and rice, they lose viability within 30 minutes from their release into the environment. So Korea, viability is nothing but doing activities or performing activity. So they loses viability within 30 minutes from their release. Viability of the pollen grain is controlled by temperature and humidity. In case of wheat and rice, pollen grains will lose as viability within 30 minutes from their release into the environment. But whereas in members of the family Rosaceae, Solanaceae, and Leguminosae, in members of the family Rosaceae, Solanaceae, and Leguminosae, they will maintain viability even months together. I will repeat this last point once again. Wheat and rice will lose its viability within 30 minutes from their release into the environment. But whereas in case of the members of the family Rosaceae, Solanaceae, Leguminosae, they will maintain the viability even months together. Rosaceae, rose family, Solanaceae and Leguminosae already we have come across in detail in PUC first year chapter Morphology of Flowering Plants. All pulses belongs to family Leguminosae. Solanaceae is nothing but brinjal, potato, all they will come under the family Solanaceae. So that ends the structure of pollen grains. Look over the last points and take care all these matters. Pollen grains are spherical in shape, measuring around 25 to 50 mm in diameter. It is covered by two walls. Outer is called as exine and the inner is called as intine. Exine is a tough, cutinized and often provided with a spinous outgrowth. This spinous outgrowth helps in the dispersal of pollen grains. Exine is having sporopollenin, one of the most resistant organic material. It can withstand high temperature, strong acids and alkalis. Pollen grains can be best preserved as an fossils due to presence of sporopollenin. Enzymes do not degrade sporopollenin. In around 60% of the angiosperm, pollen grains are released at two-celled stage. Larger is called as vegetative cell, smaller is called as generative cell. Okay, so vegetative cell is having irregular shaped nucleus. Generative cells are spindle shaped with a dense cytoplasm and a prominent nucleus. Followed by that, I covered this matter. So now take care of last point. After pollination, when the pollen grains will reach the stigma, after pollination, when the pollen grains will reach the stigma, intine germinate and produces a small tube-like structure known as pollen tube. After pollination, when the pollen grains will reach the stigma, it will germinate and produces a small tube-like structure known as pollen tube. This pollen tube will come out through the thin places present on the exine known as germ pores. After pollination, when the pollen grains will reach the stigma, intine produces a small tube-like structure known as pollen tube. This pollen tube will come out through the thin places present on the exine known as germ pore. So please remember, germ pore is the place where sporopollenin is absent. So in my next period, I will switch over to structure of anatropous ovule. Structure of anatropous ovule or Microsporangium.